Today, my guest is Professor Larry Young. He's a professor of psychiatry and behavioral sciences at Emory University School of Medicine. He's also director of the Center for Translational Social Neuroscience and the Silvio Conti Center for Oxytocin and Social Cognition. And he heads the Division of Behavioral Neuroscience and Psychiatric Disorders at the Yerkes Primate Center. He's a leading expert on the neurochemicals of social interactions and pair bonding. And this is what he's going to talk about. And he'll talk about hormones, or actually neurotransmitters in some ways, like oxytocin and vasopressin, talk about dopamine. Larry, before you start talking about how you study social interactions, in terms of what's going on in neural circuits in the brains and the chemicals involved, could you give our listeners and viewers some background on you? Where did you grow up? How did you eventually get interested in what you're doing now?
Yeah, well, thanks for having me. It's great to be able to come here and talk about our work and um, everything that we'll talk about today. I grew up in South Georgia, a little town called Sylvester, peanut capital of the world. At least that's what we say. And I grew up on a farm in the countryside. And I was always interested in animals. I always had pets. I had snakes and crows, and I had a possum that I used to ride on my head.
Huh.
And sometimes people would call me Possum Boy, because when I was probably 8 or 10 years old, I had a possum that would just ride on my head. And anyway, I just loved animals. And when it got time to go to college, I thought, well, what should I be? I decided at first I wanted to be a veterinarian. And when I got to college, I went to University of Georgia Bulldogs, and I did pretty well there. I made really good grades and everything. And I knew it was very difficult to get into vet school. I probably could have done it, but I just decided that I changed my mind. At that time, I was married. I got married on my 18th birthday, which is not that uncommon down in Sylvester. But and I thought, well, I'll just be a doctor. I'm doing pretty good in school. I can be a doctor. I didn't know too many other professions out there. I was first generation in college like that. So, but then as I learned, I had this fascination with animals and of nature, not just any animals, but natural animals and diversity in nature. And as I learned, I was a biochemistry major, and I learned about, the chemistry of life. And then I learned about genetics and the concept that, all of the instructions that are needed to build an organism are somehow laid out in that string of A's, G's, C's, and T's that can take a single cell. The egg and the sperm come together. You got a single cell zygote, and that can eventually turn into a white-tailed deer that has, certain kinds of territorial behaviors, and they know who to mate with. Or it could turn into a blue jay, or it could turn into a bluebird, or a duck, or a snake. All these different things. And so to me, that was really fascinating. And I like, as I learned more and more of the sort of the classical experiments that were done, I sort of felt, wow, this is what I want to do. I don't want to just be like a mechanic, because I felt, that's kind of like what doctors do. They see patients every day. It's a good thing to do, but that's not what I wanted to do. I wanted to be able to look in nature and to see if I could figure out, how do you get from a string of letters, A's, G's, C's, and T's, to an organism that has a behavior that they know who to mate with. The moms know how to take care of their babies. In some cases, the dads do as well. They know how to build their nests. So all that information is there. So to me, that was the mystery of life that I wanted to solve. And then, this was way before the internet. And when it got time for me to graduate, and I had done some work in a lab, working on mosquitoes and extracting peptides from the brains of mosquitoes that controlled their feeding behaviors. I knew a little bit about research. So I went to the library and looked for books about, um, you know, how do you go from biochemistry to behavior? Because I talked to my biochemistry advisor at the University of Georgia, and they said you can't study the biochemistry of behavior. He was a really old kind of old guy. He said you can study the behavior of enzymes. And I'm like, no, that's not what I want to do. So I went to the library and just looked in books, and I found this book on reproductive endocrinology. It was edited by David Cruz, and it had chapters in there about fish endocrinology, reproductive endocrinology, pheromones in fish. And then it had a chapter on lizards, and it had a chapter on snakes and reptiles, and then, rodents and primates. And I'm like, wow, this is the chemistry of behavior. Hormones are molecules, they're chemicals, and they somehow get into the brain and they bind to steroid receptors or other kinds of receptors in the brain and change gene transcription in the brain to give rise to the motivation to mate. So, I read a little bit about around that time. There was some really fascinating work being done. This was around 1989 on the biochemistry of steroid receptors and how something like, estrogen can bind to the steroid receptor, and then it gets translocated into the nucleus and binds to a specific sequence of DNA, and then that turns on genes. I thought that this is real biochemistry and it's getting to the behavior. I want to do this. And so I contacted one lab, one PI only, and that was David Cruz at University of Texas at Austin, who was the editor of that book. And he was working with reptiles, which I thought was cool. He had snakes and geckos and lizards and whatnot. And I was accepted to go there. And so my PhD project was really interesting. It was it was on these little lizards. Maybe you can see. So this was one of my first review papers, and these are two lizards having sex. These are whiptail lizards that every year I would go with other members of the lab out to Arizona desert or West Texas desert and catch these guys. And what made them so cool was there was two different species of lizards. One was your everyday run-of-the-mill lizard where you have males and females. They're in the desert. A male finds a female and he courts her. And when he courts her, that makes her start releasing endocrine hormones that makes her eggs grow. And then he has sex with her, fertilizes those eggs. There was the other species where there were no males. They reproduced by cloning. And the females, their brain was sort of transgendered at the time. I mean, one day they may think that they are a female, and another female will come and court them and do the same sort of courtship dance that males normally do. Then that other female will mount her, and they will copulate and get into this position, which is essentially having sex. But nothing is transferred between them. It's just that behavior is necessary to kick off the hypothalamic pituitary gonad axis to make the eggs grow. So it was just a behavior to stimulate this neurochemistry. A few days later, they would switch roles. The female that was behaving like a female, suddenly she would become masculinized in her brain, and then she would court the other female. So they could teeter-totter between having male sex behavior and female sex behavior. Whereas there are other species that looked almost the same. The females only acted like females. So to me, I thought that this was a very interesting evolutionary question. You've got two organisms that look almost the same, yet one has a very different behavior than the other. And my PhD thesis was about trying to, work out the details of what could be different between those two species that makes one behave in one way and another behave in another way. So that was my PhD. And so I think from now on, we'll probably talk about our work with the voles. And the reason that I started working with the voles was because I had I was very much interested in the evolution of behavior and where does the species differences come from, diversity in behavior. And I read this paper that was published by Tom Insel in 1993. It was a year before I was graduating. And in that paper, he talked about these prairie voles and a peptide called vasopressin. And these prairie voles are monogamous. Okay, so they're socially monogamous. They form pair bonds, and most of them do anyway. And then they nest with their partner. They raise the offspring together. And this paper was published in Nature showing that this peptide, vasopressin, was responsible for that pair bonding behavior in these guys. And what got me really excited was the fact that there were other species of voles that look almost the same, like the meadow vole, who not only do they not pair bond, but they're pretty much asocial. They don't have this need to interact with each other all the time. They don't have this social craving. So, when I started my postdoc, this is what I wanted to do, was to try to figure out in the brain what's different between these guys that can form bonds and very highly social versus ones that are not.
And so Tom Insel, is it at the Primate Center then?
He was so he was had a position at National Institute of Mental Health at the time, and then he moved from there right before I could join his lab. He moved from there to become the director of the Yerkes Primate Center in Atlanta. But then he left here in 2003, I believe, to become the director of the National Institute of Mental Health.
Yeah.
So he was the director of National Institute of Mental Health for a lot of years, and sort of so I worked with him while he was here, and then he left, and that was actually a good thing for me, because then it allowed me to to grow my independence.
Yeah, that's great. Yeah, he's a very important person at NIH for quite a while. That's a very obviously depression, anxiety disorders, other mental health disorders is a big problem, and it's increasing. And maybe when you get done talking about your fascinating work in prairie voles, we can talk about the implications for disorders that
exactly
some which involve so we had this COVID hit, right, and people were kind of isolated without much social interactions, and particularly young kids that may have had some impact.
Exactly, yeah. And so also disorders like autism, we can talk about that later. But Tom Insel was he was a psychiatrist, and so he came to Emory and became faculty in the Department of Psychiatry. So that's how I got into the Department of Psychiatry.
I see.
And so over the years, I really did think about social interactions are so very important, and being able to form bonds. This stuff is really, it's not just about evolution. This is about it has translational implications, and so that's why I eventually started the Center for Translational Social Neuroscience.
Now, mice and rats are the most commonly used species for laboratory research, and there's a lot of reasons for that. So why prairie voles? You mentioned this one species of prairie vole, you mentioned the prairie vole forms pair bonds and the meadow vole doesn't. Is that why you're using voles instead of mice or rats?
Yeah. But also I think if you're interested in the behavior, I mean, mice have a lot of power in the sense that they can give you the ability to manipulate circuits very easily, cell type specific manipulations, genetic dissection of behaviors. But I'm interested in natural behaviors, and the mouse has been bred in the laboratory for hundreds of generations to create a mutant behavioral phenotype, one that doesn't bite very much, one that is perfectly happy living in a cage with nothing else in there, and having lots of babies and taking care of those babies. They've been selected for this kind of behavior. So in a sense, while I think there is value in studying mice, and I'm glad people doing that, it's not a natural system. There's already been a lot of mutations that take it away from the natural behaviors.
Yeah, and they don't form pair bonds.
And they don't form pair bonds. There's a lot of behaviors that they don't do. So my goal, ever since I started, 28 years ago now, has been to try to, I like to ask questions and then find the species that is perfectly suited for that question.
Yeah.
And then try to figure out how to bring all the tools to bear to that species. And so, although it's been a long journey, now we've been able to use CRISPR to make knockout prairie voles and oxytocin receptor Cree prairie voles. And so now we have all the tools that we can do that we can use to answer some of these really sophisticated questions that neuroscience is tackling now in mice. But yeah, you're right. So the prairie voles, they, it's fascinating that when the male and the female come together and they mate. Once they mate, there's a transformation that occurs in the brain of both the male and the female that then causes those two guys to want to be together and nest together, and they form a bond. And as I mentioned, the meadow voles don't do this, and so it's a good contrast. But because social behavior is so important in our lives, social bonds, like the bonds between parents and children, are so important. The nurturing that the parents give to the children can help shape the neural circuitry of social behaviors. The bonds that we have between, our partners, but also our friends and family, are so important. So I think that the prairie vole gives us an opportunity to identify molecules and processes in the brain that are involved in this bonding process that's really relevant to us. Because after all, our family behavior is much more similar to the prairie vole than to the to mice.
And so why don't you go ahead and talk about oxytocin? That, if you go on the internet and put in oxytocin, you Google it, you'll get a lot of these random hits of people, and there's a lot of hype about it. But talk about oxytocin, what cells produce it in the brain, where does it act, both in the brain and in the periphery, and maybe start with a historical perspective, like who or when was oxytocin first discovered, and what was its first actions in terms of affecting behavior?
So oxytocin was discovered, more than 100 years ago. I mean, maybe the structure was not elucidated, but it was identified to be in pituitary extracts of animals, and it would cause uterus contractions. And so oxytocin is Greek for quick birth. So it was recognized that this is a molecule that's made in the brain, secreted from the pituitary, that when a mother is getting ready to give birth, it's pulsatile released from the pituitary. And every time that pulse of oxytocin gets into the periphery, it will go down to the uterus and cause a contraction. Okay. And later it was discovered that the same molecule is released after the mother gives birth and she's nursing her baby. And it's actually when the baby suckles the breast, stimulates the nipple, the nerve stimulation goes up to the oxytocin neurons in the hypothalamus, and then they pulse fire and they release oxytocin. And each wave of oxytocin that comes out binds to receptors on the breast cells that are holding the milk, and they contract. And this is what causes the milk letdown. And, probably over half of the women in the United States, when and one of their children receive oxytocin to help with the birth.
Yeah.
Right. So when you hear about inducing labor, That is the doctors giving augmentation of oxytocin to cause the uterus contraction. So oxytocin was the first peptide molecule to have its structure identified and to be synthesized, and it was a Nobel Prize won probably around 1955 or so for that by Vincent Duvigneau. Yeah, so there's a lot of history there. But what getting back to the neuroscience then, social behavior, it was around the late 1970s or early 80s. Court Peterson was working on rats. He's a psychiatrist in North Carolina, and he said, so oxytocin is so important for giving birth and to produce the milk. But what we also know is that when mothers give birth, if you're talking about rats, virgin rats don't like babies, right? The babies stink, they squeal, they're annoying. And you put a baby in a cage with a virgin, she'll go cover that baby up. And if it doesn't shut up, she might attack it and kill it. So there has to be some transformation that happens when a mother goes through the pregnancy. And part of that is estrogen and progesterone, the steroid hormones. But what Court Peterson thought was that, hey, this molecule that's so important for giving birth and for nursing, maybe that is also acting in the brain to cause the mother to want to take care of the baby.
Yeah.
You have to not only, prepare your body, make your body give the birth and to produce the milk and to let down the milk, but you also have to have a change in your mindset to think, oh, this baby is the most important thing in the world, and I want to take care of that baby. And what he showed was that if you injected oxytocin into the brain, you really have to inject it in the brain because it doesn't cross the blood-brain barrier very well. If you inject it in the brain, you can make a virgin female rat begin to take care of babies. Mm-hmm. And then there was other work by Barry Cavern and Keith Kendrick in England. They were working with sheep, and rats are different than sheep. That rats and mice give birth in a nest, and so they know that nest is theirs. They actually don't really bond with their individual babies. They bond with their nest. The babies can't walk around, right? So the baby rats and mice can't walk around very much. So mothers just know, this is my nest and I'll take care of anybody in here. You can give them another baby. They'll take care of another baby too. But sheep, there is a breeding season. Everybody's giving birth at the same time. There's lots of little lambs running around. So when a female becomes a mother, not only does she have to have a mindset change to say, wow, lambs are adorable. I love lambs. She has to bond with her particular lamb. She has to take in the smell of her baby and the face, actually, the facial cues of her baby. And something happens in her brain so that she says, oh, this is my baby. Then she will bond with that baby. And other babies that come around, she butts them away because she only has a limited amount of milk. Yeah. So that's a kind of a bond. And Barry Cavern and Keith Kendrick showed that you can take a female sheep who is not giving birth, but if you inject her brain with oxytocin and give her a baby, a lamb, she'll suddenly just think that's her lamb. So this chemical is acting in the brain to cause her to bond with that baby.
And she'll only bond with that baby then, and not others, or?
Well, I don't know if you give her other babies at that same time that you give her the oxytocin, she might. But once that bond is formed, then yeah, she's only bonding with that baby.
And of course, they sometimes have twins, right?
Right. So she could bond with.
Yeah.
Yeah. So it's like, yeah. So it is that oxytocin that is released when they give birth that sort of opens up a window in the brain to say, bond with something right now, because the brain thinks you just give birth, right? So the brain uses signals that are involved in other processes. And
so these neurons, the neurons that produce oxytocin, so their cell bodies are in the hypothalamus. Then they have these long axons with really big axon terminals in the posterior pituitary, and that's oxytocin is released into the blood. Mm-hmm. from there. Is oxytocin also released in a more specific manner? Are there other like branches of those axons that go
Yeah,
out other places?
yeah, those axons, they bifurcate, and then some leave that pathway that goes to the pituitary, and then they go into the brain, and
Okay.
sort of it's almost as though there's not like real specific pathways, although they're more dense in some places than in others.
Huh.
But those fibers just sort of wander through the brain, and you might see in the prefrontal cortex, for example, one little fiber coming, oh, one big fiber. These are thick fibers, and the whole fiber is jam-packed full of oxytocin in dense core vesicles. The dense core vesicles are just sitting along the axon walls. These axons are
Oh,
not myelinated. So unlike other neurons that are myelinated, they have a myelin sheath around them. These are unmyelinated, big fibers with oxytocin in vesicles all the way up and down the fibers.
oh,
So,
interesting.
so when a mother is nursing, boom, you need to have an action potential to get the milk to be ejected. Well, they also have these fibers that are going to the brain, and so that the brain, it provides a mechanism for the brain to listen to um, uh, when those oxytocin neurons are firing. And
So they're not forming like specific synapses as we know them. So it's still like a neurosecretory mechanism, but the oxytocin is being released kind of locally, and then it can affect multiple
right,
neurons in an area.
right. So there are these boutons all the way up and down the axons,
Huh, interesting.
where we don't know exactly the details of whether there's like fine-tuned control locally to make release. I suspect there is. But yeah, it's not acting at synapses. I mean, these neurons, they might have, one terminal somewhere that's glutamatergic,
Oh, yeah.
but they're very difficult to find, the terminals actually. But what you see is that these big fibers with these boutons. And so that allows the peptide to be released locally, and then you can diffuse. So one axon going through the prefrontal cortex could release oxytocin there and then bind to receptors. And so what we have realized now is that oxytocin, first of all, I've only told you about birth and nursing. Those are things that females do.
Right.
So you might think that oxytocin is a female molecule, but if you look in the brains, males and females have the same amount of oxytocin. Turns out that oxytocin is not just about giving birth and bonding with babies. It's released by social touch.
Huh.
It's released, so in studies in rats where they could put little cameras into the brain and look at those oxytocin neurons in the hypothalamus, and they genetically engineered them so that they fluoresced when the neurons were activated. They found that when two rats are interacting with each other, maybe touching their back, grooming in a positive, socially reinforcing kind of way, those oxytocin neurons were firing. Okay. Other studies have shown that oxytocin, when given intranasally to humans, causes people to look into the eyes of others more. Just like a mother looking at the baby, and like, this baby is the most special thing in the world. It also, in other social situations, it makes us tune into the social cues around us, looking at the face, looking at the muscles in your face to tell if you are interested in what I'm saying, or you're bored, or what. Extracting information from around us. Even studies now have been done in dogs and their owners to show that when a dog looks at the owner for an extended amount of time into the eyes, it causes oxytocin release in the owner.
Interesting.
And if you actually give the dog oxytocin spray, they look more into the owner, and the owner gets more oxytocin release. And I'm convinced this explains, when my wife, our children are all grown now, but we have 3 little dogs, and my wife still loves to take care of the dog. And if the dog looks up at my wife with its puppy dog eyes, it will get any treat that it wants. And actually, dogs evolved from wolves. We selected dogs, domesticated dogs from wolves. If you look at wolves that are raised exactly like a dog with the same kind of care, when they grow up, wolves don't look into the eyes of people. They don't communicate with eye contact. We somehow selected the animals that would look into our eyes and activate that oxytocin bonding system. And when they did, we would give them a bone around the campfire. And we kept doing that for thousands of years until now we have a dog that communicates like we do.
Now you've looked a lot. So which neurons respond to oxytocin? Of course, there has to be receptors on the surface of those neurons. And one thing you've done a lot of work on, really nice work, my looking through your publications, is kind of mapping where those receptors are for oxytocin and vasopressin, say in prairie voles that form pair bonds and these meadow voles that don't form pair bonds, but maybe also in, because I just thought of, has anyone looked at the distribution of those receptors in wolves versus domesticated dogs?
No one has done that. That would be very interesting.
Yeah.
But yeah, so one of the cool things that's come out of the research since very early on, we realized that although every species looked at has the same location of making oxytocin, and the fibers seem to be pretty similar across species, what's different is what parts of the brain can respond to the oxytocin. And these receptors in each different species that we look at has a sort of a unique expression pattern. And if you look at prairie voles, they are loaded with oxytocin receptors in both the prefrontal cortex and the nucleus accumbens. These are areas that are involved in reward, reinforcement learning, and even addiction. The nucleus accumbens is where dopamine acts to create reward and reinforcement, like cocaine, the rewarding aspects of cocaine. If you look at a mouse or a meadow vole, you hardly see any receptors there. So if you look at a marmoset, which is a primate, which is monogamous primates have oxytocin receptors in this nucleus accumbens. If you look at rhesus macaque, which is not monogamous, they don't have receptors there. They have receptors in other places. So from an evolutionary perspective, which gets back to my original interest of why all animals behave differently, each different species, different circuits in the brain are responsive to this oxytocin. So when they're interacting with another animal, some species may become very aggressive and territorial. Some will, potentially bond with that other animal with they mate, and it gives them all the different kinds of behaviors. And so let me just talk a little bit about the nucleus accumbens and oxytocin receptors and dopamine and bonding. What we found is that if you block those oxytocin receptors in the nucleus accumbens, the prairie voles can mate, but after mating, they don't form a bond. So you could prevent them from bonding. You don't prevent them from mating. They still mate, but the consequences of the mating are different. They are not bonded with who they mated with. Same thing with the prefrontal cortex. So this circuit seems to be very important. And we've actually been able to do some experiments where we've used optogenetic stimulation to control the projections from the prefrontal cortex to the nucleus accumbens with light. And whenever they get around one individual, we activate that circuit. When they're around a different individual, we don't. And then they show that they develop a bond. So this is the kind of cool stuff that you can do now with neuroscience and behavior.
And you're doing that in both. You're doing that in the females, and then looking at bonding with the males.
We did that in the females, but we're also doing other experiments with in males too. So oxytocin is important for male bonding as well. If you block oxytocin receptors, males also do not bond. So now we realize it's not just a female hormone. Maybe it originally evolved for reproduction to give birth. Even in C. elegans, which is a nematode, it's involved in the two animals, the male and the female, if they're the male and the females, when they come together to try to find each other's genitals, it's the sensing the other one. So it's involved. This is Corey Bargman's work. It was published in Science in 2012. So very early on, we see that oxytocin is involved in sensing the other. And what I have concluded based on all of our work with the voles is that it's involved in increasing the salience of social stimuli. One of our first clues was when we made a knockout animal. We made an oxytocin knockout mouse, and then we made a oxytocin receptor knockout mouse. We found that even in the males, one thing that was wrong with them is that they could never remember another mouse that they've met before.
Huh.
They had social amnesia. Now imagine you go to a meeting and you see thousands of people. You're not going to remember every one of those people. But if you have a real sort of an interaction with one, and pleasant interaction or even negative interaction, you're going to remember that.
Well, and you remember their face, but not their name. That's what I do.
Yes, maybe, yeah. Sometimes you can remember their name, but not their face as well. Once you go back and you see lots of people, you can remember that face, but you may not know the name.
Yeah.
You see the name tag, and then you know everything about them. Well, that's a difficult task because we all look the same. We all have two eyes here, nose here, mouth here, hair. And for mice, they tell each other apart by smell.
Yeah.
And each mouse has a little bit unique smell. It doesn't mean they have totally different molecules, but different ratios of different molecules. And so just like we use interocular distance, we still use the eyes, but how far they're apart they are, the shape of the nose, we use all these little features. Mice have to use that same information to tell each other apart, and they can remember a lot of animals. They can, and they need this for territory, who's friend, who's foe. But if you knock out the oxytocin receptors, they can't remember. And there's been now some electrophysiological work in the olfactory bulb that shows, as well as in the auditory cortex. Robert Frumke has done some great work in terms of maternal care, of listening to babies' cries, but also this olfactory input, that oxytocin alters excitatory inhibitory balance. For example, by activating inhibitory neurons that can cause silencing of noise so that the signal becomes more clear. So if you're just wandering around, people around you, there's a lot of noise happening in your brain. You're not attuned to anything in particular. But what seems to happen in mice is that when two animals meet each other, oxytocin is released. This is a social situation. If you're recording electrophysiologically in the olfactory bulb, you get like popcorn, a little bit of noise. And then oxytocin is released, the noise goes down, and the signal can come through clearly. And that's the salience of the social stimuli becomes very prominent. We pay attention to that.
So if oxytocin enhances GABA inhibitory neurotransmission, does it have like an anti-anxiety effect?
Oxytocin is very anxiolytic. Yeah. Mothers who breastfeed will tell you that at the time that they're breastfeeding, everything is cool in the world. Rats, if you take a female rat, she's not breastfeeding, you snap your finger, they jump. Breastfeeding, they don't jump. So yes, it's very anxiolytic.
That's very interesting because it, the maternal bonding and pair bonding, when you form a bond with another person, then you're kind of you're relaxed, and kind of anxiety levels go down when you interact with them. Yeah.
And so there's another molecule that I studied for a while, vasopressin, and it's very similar to oxytocin. They evolved from the same gene, early invertebrate evolution, the gene duplicated, and then oxytocin and vasopressin sort of diverged a little bit in their sequence, and they have separate receptors. And vasopressin seems to be, well, vasopressin is also called antidiuretic hormone, so it regulates your kidney function and your urination, how you can concentrate urine or urinate a lot. And but it turns out in animals, in the brain, vasopressin seems to be involved in territorial behavior and even vigilance. So whereas oxytocin is calming and bonding, vasopressin seems to be vigilant and territorial. And it's a very testosterone dependent molecule in the brain. So it's more male, typically in males, it's involved in territorial behavior. And in these voles, when they bond, sure, they show nurturing affiliative behavior towards their partner, but when another animal comes by, they show aggression and fight them away because they don't want to allow another male, for example, to come in and mate with their female and maybe get her pregnant and then have to raise her offspring, right? So,
or to, hurt the kids,
right,
the babies.
right, because they may kill his babies, right? So,
Yeah.
There's that sort of there's this yeah, the oxytocin is more calming, vasopressin is more territorial, aggressive, maybe like jealousy. I think of, in terms of the evolution of love in humans, probably humans experience love differently than other animals. Maybe other animals don't experience love. I try to reserve the word love for humans because this is a strong sort of emotional bond, but we also attach a lot of language and thought about the future and things that maybe only humans can do. But I'm convinced that the underlying sort of subcortical mechanisms that are involved in that are related to the same kinds of systems that we studied in voles, in the pair bonding and the nucleus accumbens, and then jealousy maybe evolved from this territoriality component.
Yeah, of course, it's kind of almost impossible to study these things in animals. We have language, right? We can communicate.
Right. Yeah.
And animals can't, so we can't really know exactly what they're thinking and how similar it is to what we know as love, as humans.
But we can observe their behavior,
Yeah.
and we can observe that they will take care of their offspring or bond with the partner. And so just to get back to this, the fact that the receptors in the nucleus accumbens, what I think a pair bond is that the animal, well, when they mate and they're activating this oxytocin system, the cues of the partner become very salient. The smell, maybe the sound, if they're vocal, and the visual cues possibly as well. Those become very salient. And then the animal is experiencing dopamine too. With the mating, you have the dopamine release and its reward. And I think that somehow the neural representation of the partner becomes hardwired through synaptic plasticity into the nucleus accumbens. And so that way, that partner is actually inherently rewarding.
Yeah.
And a pair bond is basically, it's very similar to an addiction to your partner. It's a very similar kind of process. And there's another aspect of bonding that we've studied recently, which is kind of interesting. It's after you form the bond, and there's a lot of dopamine released when you form that bond, the partner becomes rewarding. You seek out that partner just like maybe a crack addict would be seeking out crack, all you can think about is that partner. But that's in the beginning of a relationship. What about long-term, 10 years? I've been married 17 years now. Things have changed. And I still love my wife and I go see her every day, but I'm not like constantly thinking about her all the time. Well, we thought, let's look at these voles and see what happens if they lose their partner. Because we know in people, losing a partner often leads to increase in mortality of the remaining partner, increased cardiovascular disease, even decreased resistance to cancer. Okay. So being in that relationship is a positive kind of thing. And so what we found is that in these voles, if you take away the partner, they show depression-like behavior. And when we looked into the brain, we found that removing the partner actually suppressed oxytocin release. So they're experiencing a kind of withdrawal of the oxytocin. And then we found that if we could take an animal and when they lose their partner, immediately replace the partner with a micro infusion of oxytocin into the nucleus accumbens, the animal was no longer depressed. So there is this sort of a negative component to being away from the partner that, if extended, leads to depression, and it's just probably not very good for survival in the wild. And so, I was trying to figure out, okay, why is this? Why would an animal do this? I think that this is a mechanism of maintaining the bond. In the beginning, the bond formation requires the dopamine, the exhilaration, the linking of the cues of the partner with the reward. But after a longer period of time, it's more about when they're out foraging and they're missing their partner, then they get a negative feeling, a negative affect, and this is what drives them back to the partner. I think about, like me and my wife, when we first got married, we got a little puppy dog at the same time as we got married. And I remember every day when I would come home from work, my wife and my puppy dog would be equally excited to see me come home, but now, 15 years later, I come home from work and my puppy dog is still excited to see me and my wife, not so much. But if I don't come home, she's concerned. And if I'm away for weeks, for work, for travel in Europe or something, I miss her. So it is that negative feeling that makes me want to come back home with her. So it's sort of the yin and yang, the positive feedback. The dopamine interaction is what creates the bond in the first place, but the lack of it.
Now, so going back to the voles for a second. So the meta vole is a promiscuous species, right?
Right.
And
like
you had this paper in Nature in 2004 where you show you can manipulate the one gene and make that meadow vole be more like a prairie vole in terms of its social interactions and behavior. Could you talk about that?
Yeah. So again, this gets back to my interest in diversity in behavior and the brain mechanisms of that. And what we realized is that for both the oxytocin receptor and the vasopressin receptor, the two species have very different distributions of receptor, which means that when they mate and they're releasing the oxytocin or the vasopressin, you're activating totally different circuits. And so I thought, wow, these guys look the same. I mean, if I was to show you a cage of prairie voles and a cage of meadow voles, I don't think you would be able to tell them apart very easily. They're very similar. But their brain, it's easier to slice their brain and for me to show you their vasopressin receptor distributions or oxytocin receptor distributions and tell what species they are. So, and this suggested to me that the circuits that are responding to vasopressin, they are leading to this pair bonding behavior and this selective aggression behavior. And the fact that they're different between different species maybe explain the species differences. And in the prairie voles, we noted that, the area that's the output of the nucleus accumbens called the ventral pallidum is also part of the reward and addiction pathway. Prairie voles have a lot of receptors there and meadow voles don't. And so we thought, and if we block those receptors in prairie voles, they cannot form bonds. So we know that those receptors were important. But could you transform the behavior of a meadow vole into like a prairie vole by putting the receptors there? So it sort of gets to the idea is, can the diversity in behavior come about just by moving around the location of the expression of a single gene? So what we did was to take that vasopressin receptor gene from the prairie vole, put it into an adeno-associated virus, AAV. This was one of the first studies to use AAVs to put a gene from one animal into another. But we took this vasopressin receptor gene, put it into the ventral pallidum of the meadow voles, and then we ask, when they mate, do they have a preference for their partner, for the animal that they mated with? And indeed, we showed that they did show a partner preference. So we could transform the social behavior of one animal, of one species, just by changing the receptor distribution in the brain. And so that's why I think that in nature, these two genes, these oxytocin receptor and vasopressin receptor, are regulating aspects of social behavior. And because they're located in different brain areas in different species, they're creating the diversity of social behavior that you see out in nature. And even within the same species, you can have different animals have different levels of receptor expression. And in fact, we've looked at this in voles. So in prairie voles, some prairie voles have high levels of receptors in the nucleus accumbens, and some have lower. And that predicts how they will behave. And then, and actually it predicts whether they will be resilient to early life stress or susceptible to early life stress in terms of the ability to bond later in life. And we've found that just single nucleotide polymorphisms in the oxytocin receptor gene predict the level of expression in the nucleus accumbens of prairie voles.
And has that been looked at in humans? Are there inter-individual differences in the?
Yeah, there's a huge amount of individual variation. First of all, if you look at average amounts across different brain areas of where oxytocin receptors are in humans, one of the highest areas is the nucleus accumbens. So we are more like prairie voles and marmosets than we are like meadow voles or mice in regard to that, but not all of us. So there's a huge individual variation. So some individuals have high levels and some individuals have low levels of oxytocin receptors in the accumbens. This was done from transcriptomics, where you take out tissue, postmortem tissue, and then you sequence the mRNA there. And so there is a wide range of expression. And actually, you can relate that level of expression to polymorphisms in the oxytocin receptor gene. And we have found in 2014 that polymorphisms in the oxytocin receptor gene predict face recognition ability in humans.
Interesting.
Particularly in families where there's a child with autism. So maybe now's a good time to talk about autism, right? Because you know, I'm in a department of psychiatry. I should do something related to psychiatry, and you know, early on we realized that, hey, you know, social interactions are very important, and a lot of psychiatric disorders are caused by deficits in social interactions and being able to attune to others. And one disorder in particular is autism spectrum disorder. But you can also think of schizophrenia, where there are deficits, compromises in social interactions and trust and things like that. But many other, even, you know, like postpartum depression or depression causes changes in social interactions. But
our sociopathic behavior,
maybe sociopathic. I get that question a lot, and you know, we just don't know. There haven't been the kinds of studies in human populations to say whether sociopaths have maybe no oxytocin receptors in certain parts of the brain.
But you could see whether they have, but you know, you could get their DNA and see whether, you know, there's some,
right.
You know,
yeah, I think that has not been done, and maybe it's because you know, for those kinds of genetic association studies, you need a fairly large sample size, and I think getting the large sample size of a sociopath is difficult.
That's true.
So back to the idea of what is oxytocin doing? Okay, you said earlier, if you Google oxytocin, you will see that it's the cuddle hormone, the love hormone, the trust hormone, the moral molecule, all these kinds of crazy things. But if you listen to me, what you heard is that oxytocin, for example, alters excitatory and inhibitory balance in the brain so that social information becomes more salient. The analogy that I often use when I talk about this is if you have a television screen with some static, there's always static in the brain, and there's an image there of a face. You see the static and the face doesn't come out very clear. You don't really perceive it. But if you could turn a dial and make all the static go away, then you've got a very clear face. And that's what I think oxytocin is doing for us. And that's why, when people who do not have autism, when I meet someone, I usually look in their eyes, I look in their face, I try to extract information. Some people don't. I mean, there's a lot of variation, and maybe this related to this, how much receptors they have in the accumbens. But I'm attracted more to your eyes than your ears. There are studies in autism that say that people with autism tend to be more attracted to places of contrast, like up here, or maybe ears, or just not face, or they're also attracted to all the other things around the room. Social cues are not particularly salient. Now, I don't believe that autism is caused by lack of oxytocin. Okay. Autism is caused by a collection of genes. It's a lot of different disorders that causes problems in circuit connectivity, dynamic communication across brain areas that makes it difficult maybe to process very dynamic things. So social interactions are very dynamic. Lots of things are going on in our faces when we talk to someone. It's happening rapidly. You need a lot of cognitive processing needs to happen. And early in life, people with autism tend not to be very good at using someone's behavior to predict what's going to happen next. So it becomes kind of uncomfortable or just not very rewarding. I mean, when you can really predict what somebody is going to do with their behavior, that social interaction kind of becomes a little bit rewarding, and it's a positive feedback thing. So people with autism, for a variety of reasons, have difficulty attending to social cues, lack of motivation to attend to social cues. Maybe social interactions can be aversive. And so because of that, a lot of people have thought, well, let's give oxytocin to people with autism. Okay, if you Google it, like I said, it's the cuddle hormone, it's pro-social, it makes you hug, whatnot. So it makes a lot of sense just to give people, when they get up in the morning, they take their multivitamin, give them a shot of oxytocin too, and then they're going to go and be more social. But and so there have been studies that have tried that. In particular, one study that was published this last year, about this time last year, it was like an 8-year study. It took a long time to do the study. And they took children, they gave them intranasal oxytocin twice a day, once in the morning and once in the evening, every day for, I forget how long, but months, I believe. And along with the vitamin, and then in the end, they tested them and they found there was no improvement. And then newspaper articles around the country, said sociability hormone not effective for autism, which you could imagine people would say, stop studying it, no more funding, whatever. But if you imagine what you do when if you have a child, and let's say it does do what I say, oxytocin, if you sniff it, it heightens the salience of social stimuli for an hour or two when it's on board, because it's going to get degraded. They go get on a school bus. Now they have heightened attunement to the social world around them. What happens when an autistic child gets on a school bus and there's other kids around? It's not always a positive experience.
Yeah.
You're increasing the salience of social stimuli. good, the bad, the ugly. So they're having as much negative experience amplified as they are having positive experience amplified. So, I really think that it's not correct to think of this as just a hug drug, as something that's kind of like a vitamin, that if you just heighten everybody's awareness, then they're going to suddenly be tuned in to the world around them and much better. The one thing that works really well in autism, I mean, not as good as we would like it, but it works, is behavioral therapy, right? ABA, where you're, teaching the child social skills and using dopamine, using positive reinforcement to reinforce those skills. And then they can learn to read some emotions. Maybe it's not intuitive in the beginning, but they can kind of learn emotions from others, and they can learn some things to interact. And so that's through a therapist. And so I'm convinced that if oxytocin is going to work for improving autism, it would be in combination with the behavioral therapy, so that now you have the change in excitatory inhibitory balance. The static goes away. The therapist is there. You're making connections between cause and outcome of reward, and you're able to learn. So, and like a pair bond, if you induce, and when an animal, they mate, dopamine is released, oxytocin is released, neural connections are formed. They've learned who their partner is. That synaptic plasticity makes that partner rewarding. Weeks later, you have no oxytocin around. You're still bonded with that partner.
Yeah.
You've learned it. Well, if you use oxytocin to enhance the learning of the behavioral therapy, then cumulatively, maybe it increases the efficacy of that.
That makes sense.
So I think that that's the future. And also, one of the problems with oxytocin with the intranasal delivery is that it's difficult to get into the brain. Not much gets into the brain, right? So it's a large molecule.
Yeah.
And the idea is that some can get into the brain. But in psychiatry, there's no other example of a drug where we're giving the same molecule that's in the brain. We don't give dopamine to treat anything. We don't give serotonin. We give serotonin reuptake inhibitors. We give drugs that bind to things like the serotonin reuptake or dopamine transporter, or things that will therefore cause an increase in our endogenous molecules. So I think the future
for the classical receptors like dopamine, serotonin, glutamate, and so on, there are specific transport proteins in the membrane that remove those neurotransmitters from outside the cell, which that's very important for getting the system back so it's ready for another response. Is there any mechanisms that move oxytocin into cells?
Not in the same way as a transporter, but there are enzymes that degrade peptidases. So that's a possibility, but I think another possibility that I'm more interested in, and I think is more feasible, is somehow in evoking endogenous release. Imagine if you have a drug that you could use to stimulate oxytocin release. You give that during the behavioral therapy session, you might enhance the efficacy of the therapy. So we've been working on one, which is a melanocortin agonist, which just so happens to potentiates the firing of oxytocin neurons in response to stimuli, and it crosses the blood-brain barrier. So if we give this drug 2 vols, it causes an activation of oxytocin neurons, particularly in the context of a stimulus. And then that activates the brain's reward system, the nucleus accumbens, the prefrontal cortex, in the context of a social interaction. I don't think that this drug is a magic drug, so there are negative effects as well. But I think it's more of the proof of principle that I'm interested here, is that, another drug that causes oxytocin release is an MDA. oh, MDMA, ecstasy.
MDMA, yeah.
Yeah, sorry, ecstasy.
Ecstasy, yeah.
Yes, I mean ecstasy also releases other things. It changes serotonin. It gives the whole constellation of the high from ecstasy. But one of the things that ecstasy does is make you very pro-social, makes social interactions rewarding. So, um, but
You mentioned the melanocortin. My recollection, there's some interaction between melanocortin signaling and BDNF, brain-derived neurotrophic factor. Is there any role for BDNF in any of these?
I think that's a really unexplored area. There has not been much work with BDNF in pair bonding, but I think that there definitely could be.
Yeah.
But
so
so that's the translational aspect of the work. So
yeah,
it's very kind of I'm excited that we went from wow, the diversity in nature and evolution, and let's figure out how this works, to diversity in humans. That's what mental health disorders, and I mean, you think about many people who are on the spectrum do not consider themselves to have a disorder, and we shouldn't say they have a disorder. They're different. Okay, they process social information differently. And Temple Grandin visited here a couple of years ago, and one of the things that she said is, she is on the spectrum, but she's a professor, and she has certain unique abilities. And she said she's pretty sure that the individual who first sat around a campfire and hit two rocks together over and over and over again was someone with autism. Very sort of repetitive kinds of behaviors. And Albert Einstein, Tesla. So there's a diversity, right?
In autism, there's pretty good evidence that there's kind of a general hyperexcitability of neural networks. For example, they tend to be more prone to seizures, and actually, recording whatever EEG, and then a lot of the animal models, there's hyperexcitability. And since oxytocin, as you've said several times, seems to kind of quiet down and fine tune the signaling in the brain, it does make sense that it might have some effect.
Yeah. There is some work looking at seizures and hippocampal oxytocin receptors and involved in that. So, but I don't want to sort of hype too much oxytocin and think it's a panacea that would, treat everything in autism. I think autism is a complex disorder, and there's also cognitive deficits, social cognitive deficits.
Today's election day, and I just got thinking you may have a solution to the division in its country.
I don't know.
We can get people to, calm down, interact socially with people, and not be so antisocial. We'd be a lot better off.
Yeah, definitely. There's a lot of division in the country right now. But
so I think it covered a lot, and I hope the viewers and listeners have found this interesting. I'm going to put in links to several things. One of your longer lectures that I found on YouTube, where you go into a little more detail. Link to your book, which I failed to mention. Do you want to talk about your book on?
Yeah, if you're interested in this, the kinds of things that I've talked about here today, I published a book a few years ago called "The Chemistry Between Us: Love, Sex, and the Science of Attraction," and it's written for non-scientists, but I mean it's at a level where scientists will find it interesting as well, about sort of the chemistry in the brain and how that leads to sort of sex differences, sort of related to the transgender kinds of things that are happening now, but sexual motivation, maternal behavior, defensive behavior, territorial behavior, and sort of what creates bonds between us. So it's it's not too jargony. It is meant to be interesting and entertaining.
Okay, and I'll also put some links to some of your recent review articles, and so people can find that information quickly if they're interested in learning more about this very important area of neuroscience and the fascinating work you and others working in this area have done. I think it's really getting to the point where there is a lot of translational significance and a lot of food for fodder for even people who are who knows, drug companies may get interested in this kind of thing, for example, developing small molecules that tap in to some of these pathways for potential use in various brain disorders.
Mm-hmm. Yeah.
Okay, Larry, thanks a lot. I enjoyed talking with you. We didn't mention that you and I have very similar early life experiences growing up on farms and initially wanting to be veterinarians, but then deciding that maybe it's better to do something where you're learning new things every day and where you're advancing knowledge in an area. And it's scientific career is fantastic because you're always making there's some, it's slow oftentimes, but
Yeah.
you're always going to end up discovering new things and adding some pieces to the puzzles that we're working on.
Yeah.
Okay, Larry.
It's great being here. Thank you.
Okay. Goodbye.
Bye-bye.